Okay, so I am someone who's interested in learning about virology, and I've actually seen some things on the internet about um, viruses and the process that virologists use in order to find viruses. And so I've looked around for a while, actually, trying to find a virologist who is willing to discuss these issues with me. And Syed, who has quite the background in biology and virology. Um, you can share some of your background as well, Syed. But thank you for agreeing to do this interview with me so that some of these questions can be answered.
It is my pleasure, Jomo. Can you tell me how much time we have at our disposal?
Um, it can go up to 30 minutes to an hour, depending
Okay,
on what you have available as well.
okay, okay. So, with regards to my background, I have a bachelor's degree in general microbiology, and then I have a postgraduate diploma in general microbiology from the University of Toronto in Canada, and then went on to complete my second master's degree, which was in virology, again at the University of Toronto in Canada. Then I moved to the University of Ottawa to complete my PhD in virology. And I have been actively researching in the field of virology of viruses for the past 50 years, I would say. And in the process, I've also been a, a professor at the University of Ottawa, and I have published, I'd say, well over 200 research papers and delivered close to 380 lectures on the subject matter throughout the world. And even though I retired in 2003 as a professor, they made me Professor Emeritus, which is a lifelong title to indicate the meritorious work that I have done. And as a result, I still dabble in research. And at the same time, I founded a commercial laboratory, which is near Toronto, some 6, 7 years ago, which actually conducts work on assessing disinfectants and hand hygiene agents and indoor air decontamination technologies against viruses and bacteria and protozoa and fungi. So even though I retired several years ago, I'm still actively involved in the general area of microbiology with particular emphasis on virology.
Great. Wow. So that's quite the background you have there.
Thank you.
Mhm. Okay. So you agreed to doing this interview with me. I had shared some questions with you. Um, and so I'll go through some of those questions, and then we can follow up with anything that is relevant. Does that sound
Sure,
good to you?
sure.
Okay. So part of my interest in this subject is that like I said, came across some people on the internet. And I believe it actually has been happening since around the HIV/AIDS days. Um, but there's a group of people that question the existence of viruses. So I'm curious, have you heard about this? Have you looked into some of their critiques? And what are your thoughts on those critiques?
Okay. The only person that I have met who does not believe in viruses is somebody in my city whom I've known for a number of years. He is a highly qualified scientist and a retiree from the Canadian federal government. Other than that, I don't know anyone personally who does not believe in viruses. I'm quite sure there are people, but I don't know of any except for this gentleman that I talked about.
Okay. And as far as his thoughts about the matter, do you understand his perspective in terms of why he questions viruses?
Well, yes, and we have not had detailed discussions because I felt that even in my initial attempts to convey my point of view, there was a certain reluctance on his part to appreciate my point of view.
Hmm.
And at that point, I felt that it would not be worth my while to spend the time because my simple point is that I am a qualified and experienced virologist. I've been working with a variety of viruses, handling them, growing them, quantifying them, studying their characteristics, and so on for so many years. And uh, when somebody says that they do not believe in viruses, it jars my sensibility
Hmm.
because it is something which is uh, an attempt to deny what, to my way of thinking, is fairly obvious. Well, not obvious in the sense of being able to see something with your naked eyes. That you cannot do with viruses. But
Hmm.
the level of science, scientific research has reached a point where not only can you see viruses using modern technologies, modern mean they've been around for 70 years or so, and that you can uh, take apart viruses, put them together, study their properties um, in a very detailed fashion using scientifically rigorous methodology. So it is not airy fairy stuff. And we can talk more about it as we go on.
Okay. So yeah, that actually is along the lines of um, just so I'll give you some perspective,
Yeah,
and I understand your reluctance to have a conversation with someone who seems very sure of themselves, despite you seemingly uh, doing things that make it obvious to you
yeah.
that viruses are real. And so I get that you don't want to waste your time having those types of discussions. Um, so in terms of the critiques that I've seen out there, um, it primarily revolves around the process of isolating or just in general finding a virus in a sick person's spit or sick person's fluids. So could you go into some detail into how you go through that process?
Okay. So let me introduce a couple of terms. One of them is obligate parasite. You know what a parasite is. It's something that derives a living from another entity. So obligates are not only parasites, but they're viruses are not only parasites, but they are obligate parasites. That means that they must depend on another living being to be able to multiply and grow and carry on their life and progeny.
Hmm.
Many things can do so by relying on inorganic material. For example, if you plant a seed in a soil which has some nutrients and some water, that seed germinates and develops into whatever plant that it is capable of doing. So, but in the case of viruses, the only way they can uh, replicate or divide and grow in number is when they are brought in contact with a living host, which allows them to enter into their body and then be able to thrive by using the organic material that that particular organism contains. So,
Hmm.
if you want to, one way to isolate a virus from a clinical sample, meaning the material derived from a sick person, is to expose it or put it into animals such as mice or guinea pigs, and there are other kinds of animals, even monkeys. And if that virus is there and is alive, it then attaches to the cells of the body of that animal, enters into those cells, and then essentially takes over the metabolic machinery of that of those cells and produces more of itself. Okay,
Hmm.
this is the way viruses propagate themselves, and that is what is termed obligate parasitism. So when the virus has been allowed to grow in that animal may die, may become sick, and can also become a source of more of the virus from its own body, which you can put in a freezer or inject other animals or do other experiments with it. Luckily, some few decades ago, people were able to find that you don't have to use whole animals because there are issues of ethics and expense and so on. And also that the body of even a small animal such as a mouse is very complicated. To study something in the whole animal can be quite challenging. So people have found that you can remove cells from different parts of an animal's body and you can grow those cells in a laboratory container. Okay, this is called cell culture or tissue culture. So when you have grown those cells in your laboratory container, you can use those cells for infecting and growing and studying viruses. So the most common way of being able to do it is to use cell cultures. And over the years, people have developed dozens and dozens of different kinds of cell cultures, which you can buy from certain scientific suppliers, or you can make them yourself by taking the right kind of organ and their cells from an animal such as a mouse or a guinea pig or a rabbit. So essentially, virologists use less and less of intact animals because, as I said, reasons of expense and ethics and so on. And they rely almost solely on these cultured cells in the laboratory. For example, I have worked with easily a dozen or more types of cells derived from animals such as monkeys, rabbits, mice, hamsters, and so on and so on. And there are cells that are available that have been derived from humans. There is one particular type of cell that is extremely commonly used. It is called HeLa cells, HeLa, HeLa cells. They were derived from the cervical cancer of a lady in Chicago. So when they removed her cancerous cervix, they took apart some cells from that tissue and were able to perpetuate it in the laboratory. So you can imagine um, this being done since the early 1950s. And those cells are still very commonly used in, in research on virology. Just an example. And there are, as I said, several different kinds of examples. Now, what one needs to appreciate is that these different types of cells allow you to grow different kinds of viruses. Not all cells are hospitable to all kinds of viruses. So
Hmm.
you learn from experience that if you want to work with, say, influenza virus, what would be the most appropriate host cell for that purpose. So, for example, in the laboratory that I'm a part of, we have 15 or 16 different kinds of cell lines, which can be kept frozen at minus 80 degrees Celsius or centigrade. When we need those cells, you take out one of the ampules of the vial, put it in a laboratory container, feed it, put it at the right temperature, and the cells grow in number. And then you can use that culture to study a given type of influenza virus or HIV or coronavirus or whatever. Okay. Just stop me when I'm not making sense if I'm not making sense.
Can I, yeah, I'll just ask a question here.
Sure.
So, so you're saying that there's cells that are taken from humans or animals in these
Yes.
cell cultures,
Yes.
because and initially you had to use animals or humans, but moved away from that
Yes.
because of ethics. But the the thing that you said about using those animals and humans is that they were in living creatures. So
Yes.
how can you, I guess, equate what's happening in the cell culture with
Mhm.
those cells to what would happen inside of the animal or the human? How do you know that those are similar processes?
Okay. So, good question. So, supposing I have isolated a particular influenza virus from one of these cell cultures in the laboratory, I can take that virus, which came from the cell culture, put it into an animal, such as a ferret, and show that that culture grown virus can infect that animal and produce influenza. Okay. So
So
this,
you have done that?
Yes, yes. And this is the normal way people do it. You, um, again, there has to be a justification for the use of animals unless
Hmm.
and until you can satisfy the ethics committee that what
Hmm.
you're doing is totally scientifically valid. If there is an alternative, one chooses to use that rather than sacrifice one or more animals. But
Right.
this can be done, has been done, and is being done on a regular basis. This is especially the case when you are developing a vaccine. So, what you do is to develop your vaccine in a cell culture, but how do you know that that vaccine will actually work in an animal or a human being?
Hmm.
So, take that vaccine, put it into an animal, let the vaccine develop antibodies against that virus, and then challenge that animal with a hot virus and show that the vaccinated animal are the ones that are protected from the infection. Okay. So this is
Okay.
done on a regular basis, and that has to be done to be able to satisfy the requirements for registration of a given vaccine for use in animal or in human beings.
Okay. So going back to finding the virus in the cell culture,
Yes.
um, so I have two questions about that. One, have you ever found a virus without having to go through this cell culture process, just in the fluids of the sick person?
Yes. Um, that is quite a common practice. One good example is that of viruses called noroviruses. I don't know if you've ever heard of that term, or rotaviruses, ROTA viruses.
Yeah, I've heard of them, but I don't know too much about them. Are you
Okay,
saying
so
that they are viruses straight from the spit?
yes, so they cause gastroenteritis or diarrhea. So if you have a patient who's suffering from acute diarrhea and you want to know what is the causative agent, one of the things that you would do is to take a fecal sample from that patient and put it under an electron microscope. Okay. And if there is a rotavirus, for example, these are round viruses, quite distinct in their appearance, and they're found in such large numbers in fecal material that they become easy to look at in a fecal sample.
Hmm.
Right away, you can make a direct laboratory diagnosis of the fact that that particular patient is suffering from a diarrhea caused by a rotavirus. Okay. And
Okay.
you do,
So
you,
is it kind of like a
well, much
bacteria and a
smaller
virus?
than bacteria.
Okay.
Viruses are by their very nature nearly 1,000 times smaller than ordinary bacteria. This is why you need an electron microscope, not
Hmm.
an ordinary microscope, because an ordinary microscope simply does not have the power of magnification to allow you to look at viruses. You need an electron microscope. There are
Hmm.
other test method that one can use if you don't have access to an electron microscope. But to answer your question about being able to visually observe a virus in a clinical sample directly without using any cell cultures of animal, I give you one example. There are many such examples.
Okay.
So a good clinical laboratory in a hospital would have access to one or more electron microscope with people trained to be able to use those instruments to analyze clinical samples.
Okay. So once that has been identified in the sample,
Yes,
are you able to extract it
yes,
directly from the sample?
yes. So what you do is to use techniques such as ultracentrifugation. You know what a centrifuge is?
I think I've heard of it. It's where it like shakes and vibrates. Is that and
No,
it has like different layers.
no, it
It's like
actually spins,
okay,
and ultracentrifuge can spin at a very, very high speed, several thousand revolutions per minute. So the higher the speed of spinning, the higher is the G value, gravitational value. So for
okay,
something which is very small, if you want to remove it out of a liquid, you put it in a tube in an ultracentrifuge and spin it. By that force of spinning, even smaller particles such as viruses go to the bottom of the tube where you can then scrape up what is there and look it under the microscope. This is one way to do that. The other way is to put that sample into a cell culture, put it in an incubator, and wait for 2 or 3 days or 4 days, doesn't matter, and then look for changes in the appearance of that cell culture. Okay. If there is in most cases anyway, if there is a living virus in that sample, the cells in the culture will undergo degeneration. That means that they would begin to become sick and they will begin to die. And that you can observe in an ordinary light microscope. You don't need uh, a, uh, an electron microscope for that purpose. Okay? Just by
okay,
looking at that culture that you have inoculated or exposed to your clinical sample, you can tell not only that this particular sample had a virus in it, but also looking at the type of degeneration, you can make a presumptive diagnosis at the type of virus that might be there. Because a rotavirus produces different kind of degeneration compared to an influenza virus, compared to an adenovirus or whatever. So this is a uh, faster and a rapid and um, a more direct way of being able to detect the presence of viruses in a clinical sample.
okay. So you take the virus either from the clinical, or I mean, it usually is from some kind of clinical sample, and you
Yes,
put it in the cell culture, and then
yes,
you observe the effects that
yes,
that has on the
yes,
sample. So
yes.
what else is in that cell culture besides the cell and the sample? Is there any other ingredients in that cell culture?
Yes. So that cell culture requires a nutrient medium. It's like trying to grow a plant. You cannot grow a plant without the right kind of fertilizer uh,
Hmm.
to allow its roots to germinate and for the plant to grow. Similarly, A cell culture or the cells in a culture are exposed to a nutrient medium, contain several kinds of amino acids and vitamins and a little bit of human serum and some salts and so on. And the composition of such a culture medium has evolved to a very sophisticated level over the past 60, 70 years. And so such media are commercially available. You don't have to make them yourself. You can buy them from many laboratory supply houses, and they are quite well characterized and reliable in being able to support the growth of your cells, which will then support the growth of whatever virus you're working with.
Hmm. Okay, so it kind of keeps the cell alive, is what you're saying?
It not only keeps them alive, but induces them to divide and increase in number. So if you started out with 10 cells in the right nutrient medium, in a couple of days, there might be a few hundred of those cells there in that medium.
Oh, okay. So then when you take the virus and you put it in that medium, are
Right.
you saying, so what usually do you see happen when you do
Okay.
that?
There is a very intricate but predictable pathway that occurs. When you put a virus into a cell culture, the virus, by its very nature, attaches to the outside of the cell, and it eventually then works its way through the outer layers of the cell, enters into the inside of the cell, into what you know as cytoplasm, which is the jelly-like material that is inside a given cell. And once it gets there, it releases certain signal which eventually allow the virus to take over the cell's metabolic machinery. Okay? It simply usurps the metabolic machinery of the cell, and it redirects the cell to produce more of the virus's proteins, DNA or RNA, and other chemicals. And within no time at all, that mechanism results in the production of many progeny viruses. From one virus, you might have 100 or more viruses being produced inside that cell. And when that process of virus production inside the cell reaches a certain level, the cell becomes so sick that it actually bursts open, releasing new progeny viruses identical to the parent virus that you put into the culture.
Okay, and then do you see this happen in all of the cells that had been
Yes,
replicated in the sample?
yes. If you put them under the light microscope, you can follow this process of degeneration over a period of several hours. And as
Okay,
I
so it
mentioned
affects
before,
every cell in that
it affects
cell
every
culture.
cell. The entire culture is then destroyed after a few hours or a couple of days. And as I mentioned earlier, the type of degeneration that a given type of virus produces in those cell cultures allows you to make a presumptive diagnosis as to what virus you're dealing with. Because an influenza virus will produce a different kind of degeneration, which you can see under the microscope, compared to a rotavirus or other viruses. And
Hmm.
this, I want to introduce another term here. This is called cytopathology. Cyto means cell, pathology is damage. So cytopathology can be different depending on the type of cell and the virus. And with some experience, you can tell as to what virus it is that you're dealing with.
Okay. You can tell just by looking at it, or
Just by looking at the damage that is being caused under a light microscope.
okay. So how about the biochemical structure of that virus? Is that ever looked at to
Yes,
determine what virus it is?
yes. So to be able to do that, you have to first produce a large enough quantity of the virus you're working with, right? And once you have done that, you can concentrate it and then use that concentrated virus to be able to study its biochemistry. You can study its lipids, you can study its protein, you can study its nucleic acid, and any other component that that virus may have. Now, there are, and again, I want to introduce two terms. In a broader sense, viruses come in two types of structures. One is where the virus is surrounded by a fat containing membrane. Okay, I'll explain a little bit more later. The other category is where viruses have nucleic acid and proteins, but they have no lipids or no fatty material. Okay? For example, if you look at HIV, it has an outer envelope which contains fatty material. Okay? You can see this under the electron microscope. But if you look at the virus that we talked about earlier on, a rotavirus, it does not contain any fatty material or very little fatty material. So the viruses that contain this fatty envelope, fatty material, are called envelope viruses because that fatty material is like an envelope which surrounds the core of the virus. But there are other viruses which are non-envelope. They don't contain any fatty material or this second coating to them. There
Okay.
are huge practical implications for describing these two kinds of viruses. Viruses that contain this fatty material or viruses that are enveloped by their very nature more fragile. They don't survive as long in nature, and they're also easy to kill by even ordinary soap. Excuse me, because the fatty material reacts with soap and it can break it down. And once that happens, that virus with the fatty material is dead. It has no way of being able to infect anybody, human beings or animals or cells. Whereas those viruses which do not have this fatty layer can survive longer in nature in general terms and also are more resistant to disinfectants and so on. So this, as I said, has practical implications.
Okay.
Okay.
So I do have a question going back to the process of um, taking the sample, putting it in the cell culture.
Yes.
Um, so you've done that with supposed sick samples, like people that are sick, right?
Yeah.
Have you done that with healthy
Yes.
healthy samples and compared the results?
Yes.
Okay, and what have you found?
So this is a standard practice in virology where you use what is called a control, which is a clean sample, compare it with a positive or a known clinical sample. So when you would infect cell cultures, you have two types of cultures. One that are exposed to the material from uh, someone who's not sick compared to cultures exposed to the material coming from a sick individual. And you keep them under identical conditions in the laboratory and you observe them for this degeneration that I talked about. Okay? This is essential to be able to come up with any conclusion to say that your sample that you're looking at is indeed positive for a given virus. You have to run that control. Without it,
Okay,
your testing means nothing.
I agree. So that's great to hear that you've done those control experiments yourself.
Yes. Yes.
Um, do you have any links to those types of experiments? Because I know that a lot of the people who are questioning this whole process, they claim that no control experiments are done.
That is not entirely true. It is sad to hear that because this human pathogenic viruses were discovered in something in 1902. Okay. Animal pathogenic viruses discovered even before that, and plant pathogenic viruses even before that. So there are viruses that affect plants, animals, and humans. And there are some viruses which can affect both humans and animals. So over this 120 years or so, there has been an extensive amount of research that has been done where dozens and dozens of different kinds of viruses have been isolated from sick individuals, human beings or animals, and have been isolated either in animals or in cell cultures in the laboratory and have been characterized to a very intricate or close level where not only do we know the precise nature or chemical nature of a given virus, the differences between different kinds of viruses, but we also are able to study in great detail the structure of these ultramicroscopic entities. And let me tell you that when you look at a virus particle under the electron microscope, under an electron microscope, you're absolutely intrigued, amazed at this beauty that it entails, even though it is you're unable to see even in a regular light microscope, but they are architecturally wonders of nature. They are intricate, they are beautiful. In fact, you know what a geodesic dome is?
Yes.
Okay. So it is said that the inspiration for the geodesic dome by Buckminster Fuller, the American architect, was derived from the structure of certain types of viruses. I
Wow.
mean, I as I said, I work with viruses regularly. I don't look at them under the microscope on a regular basis because it is a bit too time consuming. But when I do it, I'm spellbound by the beauty, the intricate nature of how viruses are put together. In fact, we haven't talked about viruses that infect bacteria. There's an
Hmm.
entire family of viruses that infect bacteria. These are called bacteriophages. It comes from two words, bacterio, which is which stands for bacteria. Phage is to eat. Um, so these bacterial viruses actually eat bacteria.
Hmm.
They're again marvels of nature. Their structure sometimes is so involved that they look like a miniature satellite. Satellite with its head, its tail, its fibers, and so on. Unfortunately, I don't have a model here with me. I have it somewhere else where this is depicted in great detail as to what one type of bacteriophage might look like. Okay, so let's not live in this false impression, and I have heard this said by somebody that oh, viruses are no more than dots, they're no more than particles which are amorphous. Completely, totally wrong. Okay. If only one way to look at a good book in virology, they will appreciate how intricate these so-called dots can be. Okay.
Hmm.
And
Right. I understand when you look at it very closely, what appears as a dot can have, all of these amazing characteristics that you're talking about.
absolutely.
So I guess the main question at this point is, so you found these viruses in these samples. How can you prove that, I mean, until you show that these particles can actually make someone sick or an animal sick or something like that, it's how do you make the leap from saying, okay, this is a particle to this is a virus?
Okay. Um, well, there is such a thing as Koch's postulates. Robert Koch was a famous German scientist, and he developed these postulates, which suggest that to prove any relationship between a virus or a bacterium and the disease, you have to do certain things. You first identify patients which are suffering from that particular clinical condition, say HIV. You must isolate the causative agent that is responsible for producing that disease. You must grow it in the laboratory, and then you must take the material that you have grown in the laboratory, put it back into animals or humans if you can, and reproduce the same disease. This is the crux of Koch's postulate. If you cannot satisfy these postulates, then you cannot establish a relationship between your infectious agent and the disease that you are studying. So Koch's postulates have been utilized in establishing this relationship between a given virus or a given bacterium and the disease that it's supposed to be incriminated in. However, there are certain situations where Cox postulates are difficult to prove because our sophistication in terms of scientific research has not reached that level. For example, there are certain kinds of cancers which are produced by viruses, but you have a difficult time in producing those cancers in animal. But even if you have successfully produced that cancer in an animal, it is even more difficult to then produce that cancer in human. One is for ethical reason, and even if you were daring enough to do that experiment, you may not succeed. So at the moment, there are certain examples where Cox postulate cannot be fully satisfied. But in the vast majority of cases of an infectious agent and a disease, Cox postulates have been satisfied. So to go back to your uh, question, you have to invoke Cox postulate as the first step in being able to link a given virus to a certain type of disease
Hmm.
infection.
Okay. So the going back to the steps, I've actually never heard Cox postulates include the cell culture part. What I've heard Cox postulates is you find a potential pathogen, you isolate it, and then you run that experiment on, to see if that isolated variable is responsible or will lead to a disease state in a human or an animal. So have you ever seen with viruses Cox postulates used in that way without using the cell culture, but straight from the the sample
Yes,
and then proven to cause um, illness or disease?
yes, because that was the only way before the advent of cell cultures to
Mm-hmm.
prove the Cox postulate, because you did not have access to or the capability of uh,
Okay.
infecting cell cultures.
But now that cell cultures exist, what I mean is, have you done this experiment even though cell cultures do exist, but still run that type of experiment?
no, not I have not done such an experiment.
So the issue that um the group that I'm referencing in this interview has is they claim that what is happening inside of the cell culture is producing the particle that is then labeled a virus. And so, in order to kind of debunk that, the only way I think would be to take it directly from the sample, take the particle directly from the sample, and then prove that that particle, without using any sort of cell culture process, that particle is pathogenetic.
Well, that is an argument that can be addressed simply. I don't have the facilities or the resources to be able to do that, but it is um, amenable to testing that hypothesis quite simply by taking a clinical sample or more than one clinical sample and then exposing susceptible human being or animals to it to reproduce that particular type of infection. I don't see any impediment in that, but um,
Do you think that
again,
there's a chance that what's happening in the cell culture
no,
doesn't reflect what happens inside of an animal or a human.
No, because as I said, we run all kinds of controls. We
Mm-hmm.
run these experiments with large enough numbers of replicates. We run these experiments not once, but several times to be able to establish that relationship between a particular virus and a particular type of disease. Okay.
Hmm.
And this is, as I said, I'm not a clinical virologist, but people who work in clinical virology do this on a regular basis before they can label or incriminate a given virus in the causation of a particular type of infection. It is essential before the scientific community would pay any attention to it. Okay. So
Mm-hmm.
that is how links have been established between given types of viruses which infect animals or humans or plants and the kind of infection that they are capable of producing. There's no question. Let me just um, take this one step further. You have heard of the disease called smallpox?
Yes.
Okay. Smallpox was a truly horrible disease caused by a particular type of virus. But a few years ago, in the 1960s, 1980s actually, that disease has been wiped off the face of this earth. Now, this I want to repeat, wiped off the face of this earth. Now, this is, by anybody's standards, a dramatic event and an achievement of medical science that they should be proud of, because smallpox was a horrible disease. It killed hundreds of thousands of people every year. Those who survived were left with ugly scars on their face and on their bodies. Sometimes they were blinded by the infection. And it was so common and would occur with such regularity that parents did not dare count their children as their own till they had survived an outbreak of smallpox. Okay? So this disease has been wiped off by the use of vaccination against it. Okay, so the detractors of vaccination should take note in that vaccines have done marvels for human health and even animal health. So when the World Health Organization, sorry, go ahead.
No, I was just gonna say, like, I feel like that's a little off topic, but I wanted to like bring it back to this that we're talking about, the process, the process of determining that a virus is actually a virus. Um, so the control experiments that you're talking about, I I think everyone that I've met online that that believes that viruses don't exist would love to see those actual control experiments. Do you have any links to studies or anything that you could send me from your work or from any of your colleagues' work for the control experiments to basically show the result of the first side of the control experiment and the second side?
Yes, I will try and dig that up because, as I said, I'm not a clinical virologist or a clinical microbiologist. So I'll have to do some searching to send you what I would consider as good examples of that phenomenon that we are discussing. So the control experiments require that the cell cultures that you use to start with are free from any viruses. Okay,
Hmm.
because some kinds of cell cultures are inherently infected with viruses. So you don't use those in your control experiment or in the study of clinical virology. So that is one of the preconditions. And so when you don't see anything in your control cells which have received a, a dummy sample, that serves as the basis to be able to follow up the infected cells with their degeneration. And you can repeat that experiment once, twice, three times to make sure that your isolated virus, in fact, is able to produce that cytopathology.
Okay. And so the ingredients in those two experiments, the one with the sick sample and then the healthy sample or the dummy sample, as you said,
Yeah,
they have the same ingredients in the cell
same
culture, correct?
ingredient, treated identically, incubated at the same time for the same length of time, done by most likely one individual who is a laboratory technologist under conditions where there is virtually no chance of any virus that may be floating through to infect your cultures. This is done under elaborate biosafety cabinets, which filter out the air to exclude any contamination that might accidentally enter our cell cultures.
Okay, that's great. That is definitely something that I have not heard of. So I would love, I think that would be amazing if you could produce those results, or someone that has produced those results, if they have the write-ups on that, or if they publish any kind of papers on that.
Well, you said
Yeah.
you're in Tucson, Arizona.
Yes.
Ah, so a thought occurs to me. You should contact a very well-known microbiologist who's also a virologist who is at the University of Arizona in Tucson. Would you be amenable to do that?
Sure.
He's a renowned environmental microbiologist who has huge experience in working with viruses and bacteria and fungi and so on. He's a good friend of mine, and I will send you his contact details. And I would highly recommend that that you get in touch with him, and in fact visit his laboratory where you'll
Okay,
be able to see this whole process in action.
that would be
He,
great.
yeah, he works on um, mostly environmental sample like sewage and water and air and environmental samples and so on. But he's one of my favorite researchers, and he's extremely highly accomplished. So
Okay.
it's
And he runs these control experiments. Is that what you're saying?
well,
Or
this you'll have to talk to him about it. If he himself does not do it, he may have colleagues that do that.
okay.
Okay, so but he will give you a very in-depth perspective, again with the aid of some props or some cultures that you may be able to actually see, okay, which I can't show you. I wish I could. If you were in Toronto, I would invite you to come to our laboratory and see how we isolate viruses, how we quantify them, how we uh, study means of their transportation in indoor air, and so on and so on.
Hmm. Okay.
Okay.
Well, maybe at some point that might happen. I'm not sure if I'll be in Canada anytime soon, but
But I think your
yeah.
best bet would be to contact this gentleman that I will send you the name and contact address on. And
Okay.
hopefully he will be open enough to invite you to his lab. Okay.
Okay. Well, thank you for offering to recommend him to me. Um, so in terms of everything that we've covered so far, let me see if there's any other last questions. Um, so I feel like you've covered everything actually at the moment, but if there are any further questions, I will email you or would you be open to a follow-up interview if that's necessary?
Absolutely. Yes. Yes. Because
Okay.
my objective in my retirement is simply to make sure that if I can be instrumental in convincing these naysayers into recognizing that viruses do exist, I would be very happy.
Okay. Well, that's interesting that that's your objective. And
Yes, sir. Because you
yeah,
feel as if you have wasted your life when somebody raises this issue to say, have I been blind all this time? Have I been misled all this time? Or the other party is they are not open minded enough to say we're talking about scientific research, which by its very nature demands controls, demands a certain strict routine before being able to make any pronouncements or publish a peer reviewed paper. There is a whole set of um, do's and don'ts and controls that have to be invoked before I can come out and publish a peer reviewed paper.
mhm. Yeah.
So,
So that's why I would love to see any control experiment papers, because that is one of their biggest um, arguments is that there are no control experiments done in this process. So
they
yeah,
are, as someone has said, that they are uh, forgetting the forest for the trees. There is plenty of evidence. One has to be open minded to be able to accept it. If you're close minded, an elephant might go by and you say, well, do elephants exist? This kind of negative approach to it does not help.
I agree.
Anyway,
Yeah, I
so
think I think just completely throwing it out and not questioning virologists to begin with, I think that's the problem. And it has been frustrating, to be honest. Reaching out to virologists, asking these types of questions, I don't get very much response. And so
because to a
I
virologist,
appreciate you for being willing to discuss this.
no, I'm certainly happy to do it. And as I said, do come back with follow-up questions. My knowledge is limited, obviously, but I can at least point you to certain resources that may be readily accessible, such as this friend of mine in the University of Arizona in Tucson. Okay.
Great.
Okay,
All right.
Jomo.
Well,
Good
thank you so
luck.
much for your time, Syed.
No problem. Bye
All
now.
right.
Have a
Take
good
care.
afternoon.
You too. Bye.